Welcome to the Blueprint of Life video 7. In this video, we're going to be looking specifically at monohybrid crosses, which is the first part of Mendel's work. And we're going to be looking at how this work was used in a variety of ways. And this lesson specifically focuses on quite a bit of terminology. So first we'll start with some meta language. And when we refer to something as monohybrid, we're talking about an individual that has contrasting features for one characteristic. So when we discuss monohybrid inheritance, it's the inheriting of a single pair of characteristics. On the other hand, and which not in this list, is dihybrid inheritance, which is where we can investigate two pairs of characteristics. And then we could look at dominant, which we referred to in stage 5 biology course, where something is showing the same trait as the parents and it's represented by an uppercase letter. And on the flip side is recessive, which is contrasting to dominant and represented by a lowercase letter. We refer to genes throughout this video, and they were sometimes called factors by Mendel, and they pass from one generation to the next. And alleles are contrasting forms of the same gene. When we refer to something as being homozygous, it means that both copies of the gene in a cell are the same, so they have identical alleles. And if we refer to something as heterozygous, the alleles of a gene are contrasting or differ for the trait. So here is a quick explanation of a monohybrid cross and the outcomes that Mendel achieved to, make this something that is common knowledge now in the scientific community. So what Mendel originally did is that he had a monohybrid cross for the height of plants. So he bred a purebred, so a homozygous tall plant, with a purebred short plant, which is also homozygous but homozygous recessive. So the tall is homozygous dominant, short is homozygous recessive. And we can see here that the law of segregation tells us that each allele is inherited separately, so that all of the gametes from the purebred tall will contain the factor T, and all will contain the lowercase t, so that all of the F1 generation, so the first generation, all of the individuals there inherit one factor from each parent. So in this case, that all of the offspring are tall, but because they are heterozygous, which means they have a dominant and a recessive allele, that they'll be displaying the dominant trait, which is tall in this case. Here we have F1 generation, so the first generation are performing a monohybrid cross. And here again, we see the segregation of the alleles. But the resulting generation, so the F2 generation, in fact will show a wide variety of characteristics. So we see that the resulting ratio is 3:1. So we see 3 tall plants and 1 short plant for every 4 plants that are produced. But in fact, what is happening and what we are seeing is that out of the 3 tall plants that we see, 1 is homozygous and 2 are heterozygous, and then we see 1 short, which, because it's displaying both recessive alleles, is going to be short. And so for a monohybrid cross for the F2 generation, Mendel told us that the ratio is 3:1, and that's the ratio in which we use today. Alternatively, and what we've used before, is a Punnett Square, which is also an alternative way of representing this cross that Mendel performed. And we can see here that out of the 4 possibilities here, we have a 3:1 ratio. So, just to explain a little bit further about this model of inheritance, Mendel proposed that each characteristic or trait in an individual is controlled by a pair of inherited factors, and that in fact these factors are passed to successive generations as unmodified genes. Individuals have 2 factors for each characteristics, and that they can either be the same or different, and remember the same homozygous, different heterozygous. The trait that is expressed or which appears in the hybrid individuals is dominant, whereby if there is 1 dominant allele and 1 recessive allele, that recessive allele will be masked, and that during gamete formation, the pair of factors for a trait segregate so that they separate, and we notice that when we do the crosses so that each gamete receives only one factor for the trait from each of the parent generations. Here are some links to 2 videos which might help explain this further, but we will cover these in class. Thank you for watching video 7. Make sure you tune in for video 8.